In this part of the video, we will be starting the new chapter, that is the membrane potential and the action potential. So in this part, here we have the basics and the resting membrane potential. In the further videos, we will learn the action potential in the neurons. So first, here we have, let's imagine that is there is selective permeability to potassium only. When the potassium is going from inside to outside, that is when potassium is high in concentration and in the inside, and outside it is less in concentration, that is it's creating a negativity inside. This negativity is almost 94 millivolts. And another situation where sodium is coming out, sodium is going from out to the inside, that is sodium is outside more and inside it is less. So here the EMF, the electromotive force here would be 61. Within a 1 milliseconds they stop due to diffused potential diffusion potential. So this is calculated by Nernst equation or the Boltzmann equation. We should know the signs, how we are giving signs to them. So for positive signs, it is the diffusion from inside to the outside, and for the negative sign, we are giving sign from when the diffusion is from outside to the inside. The first is the Nernst equation. The Nernst equation is calculated by the electromotive force which is equal to plus or minus 61 upon Z. The Z is the charge for the ion. For example, there is plus 1 charge for potassium, and multiplied by the log of concentration inside to the concentration outside. Taking a hypothetical example where inside is 10 times more than outside the potassium. So K here will be plus 1, concentration of inside will be 10, and outside is usually taking 1 or 0. So log of 10 comes out to be 1, and 61 into 1 will be give 61. That is the electromotive force for potassium here will be 61 millivolts. This is the hypothetical answer. The limitation of Nernst equation is we can only analyze one ion, whereas in action potential we know that there is sodium, potassium, and chloride also being used in. So the limitation is only one ion can be analyzed. The next is the Goldman's equation. In Goldman's equation, the EMF is minus 61 into the log of C and P. The C is the concentration and P is the permeability of sodium, potassium, and chloride upon the inside and outside once again. So in Goldman equation, again we check the polarity, the concentration, and the permeability. With this, the examples can be taken as the neurons, the nerve cells, and even other cells. Here, the voltage is directly proportional to the permeability. Then, what about the electronegativity? If chloride is coming from outside to the inside, obviously inside it is negative it is negative, so inside it will cause more electronegativity. If sodium is going from inside to outside, then again inside will be more negative. And fourth, we understand from the Boltzmann equation that the sodium and potassium pump permeability will be higher than the chloride permeability. So there are several cell types and their resting potential. That is, the neurons will have minus 60 to minus 70. Generally, minus 70 millivolts is being taken. Skeletal muscles minus 85 to minus 95. Generally, minus 95. Smooth muscles minus 50 to minus 60. Cardiac muscle minus 80 to minus 90. Here, astrocyte, erythrocyte, and photoreceptors have the other resting potential. The important ones are the neurons, skeletal, smooth, and cardiac muscles. We should remember them. So the resting membrane potential of the neurons, that is minus 70, it requires the active transport of sodium and potassium pump. We already know that in sodium potassium pump, 3 sodium is taken up outside of the cell and 2 potassium is taken inside of the cell. This will create an active transport. And even we have the leaky potassium channels present. So here, this is the sodium potassium pump and potassium leak channels, the tandem pore channels. These are also known as tandem pore. So the finally normal resting potential will require three things. That is the diffusion of sodium and potassium and sodium potassium pump itself. inside the cell it will be minus 90 millivolts. Then here in first and second case there is diffusion of only potassium. So the inside is minus 94. In case of second picture we have diffusion of sodium and potassium which leads to minus 86 millivolt. So finally in the origin requires sodium and potassium pump, sodium diffusion and potassium diffusion. Next, in the next video, we will learn about the action potential in the neurons.